Thank you everybody for joining today. Good morning, good afternoon, or whatever time it is. I think maybe the prize for the furthest person away is I see one of our new faculty, Paul Dalton, joining us from Germany. So Paul, good to see you. So this lecture is part of the Night Campus Entrepreneurship Speaker Series, and I'm really pleased to welcome our guest, Professor Jennifer Elisseeff from Johns Hopkins. As long as I've known Jennifer, she's been really committed to translating her research in regenerative biomaterials, and I think she'll tell us today about some of her entrepreneurial activity as well as her science. So before we get started, I'd just like to remind people how we're doing this, a little different than our normal lectures, of course, since we're on Zoom. Please stay muted through the whole presentation. And if you have questions, which we certainly welcome questions, send them to me in a private chat and I will call on you. You just need to say you have a question at the end of the presentation. I will call on you to ask Jennifer her question. All right, so just a little bit of an introduction then. Dr. Elisseeff is an extremely accomplished individual. She is the Morton Goldberg Professor and Director of the Translational Tissue Engineering Center at Johns Hopkins Department of Biomedical Engineering and the Wilmer Eye Institute. She has appointments in chemical and biological engineering, material science, and orthopedic surgery. Jennifer was elected an institute of the American Institute of Medical and Biological Engineering, the National Academy of Inventors, and is a young global leader selected by the World Economic Forum. In 2018, she was elected to both the National Academy of Engineering and the National Academy of Medicine. And Jennifer, I'm not sure if I've had a chance to congratulate you on those accomplishments, but that's absolutely amazing.
Thank you.
Jennifer received a bachelor's degree in chemistry from Carnegie Mellon University, PhD in medical engineering from Harvard MIT Division of Health Sciences and Technology. She then later did a postdoc at NIH. She's extremely well published with over 200 papers, book chapters, and patent applications, and received a number of awards along the way that I won't list all of these to give her enough time in her talk today. Her research is focused on the development of biomaterials for regenerative medicine with applications in orthopedics, plastic and reconstructive surgery, and ophthalmology. And she's now most recently moved to an area that has a lot of interest to me, which is biomaterials mediated regenerative immunology and the role of the adaptive immune system in tissue repair. So with that, I'll turn it over to Jennifer. She'll give us a lecture today, and the title is Lessons in Translational Regenerative Immunology and the Role of Technology Transfer in Guiding Discovery. Jennifer, thanks so much for joining us today.
Thank you, and thanks to everyone for joining me. I will stop actually at a few points during the presentation to see if there either through the chat or Bob, if you want to verbally list any of them out, that would be great. So I'm excited to talk to you about how we've tried to weave together the translation process and really show you that that translation process was critical for defining new areas of research. So we often think of technology translation as being important for putting our research out there to help people, but it also goes the other way around. So that translation process really helps us understand, are we working on the most therapeutically relevant things in the laboratory, right? So as Bob mentioned, I work in the area of regenerative medicine. So although organ donations have increased over the years, it's still far below the needs. And of course, we can't transplant all the tissues that we need to be repairing. One example that I'll be talking about is the knee joint, osteoarthritis, where the current treatment now is still synthetic implants. And then I'll show some examples with breast implants and sort of new understanding of the immune response to these synthetic materials. So as you all are aware, the field of tissue engineering really developed, let's say back in the in the 80s. And the general paradigm is that you have this three-dimensional biomaterial that is designed to serve as a framework for cells and to proliferate, develop new tissue. We can incorporate biological signals. And when I started my laboratory, stem cells were just becoming a real focus of research. And so this was a really fun target to look at for biomaterials design. How can we put the right signals in these scaffolds to get stem cells to build new tissues? But at the same time, clinical translation was slow. So the two areas that we started out 20 years ago was the stem cell part, but then also looking at how can we make a clinical impact sooner, right? So the field had already really made these grandiose promises of how we could improve clinical care for tissue and organ replacement, but it was slow in making impact. And I thought we needed to do something about that. So from the technology platform side, we worked in the area of hydrogels. And we had a base hydrogel material that was synthetic, so polyethylene glycol. And onto that base material, we would put various biological cues. So we looked to the normal matrix that the cell type sees. So in cartilage, that's hyaluronic acid, collagens, chondroitin sulfate, and we tried various methods of peptides, proteins, full molecules, of putting these signals into the synthetic materials. And we looked at multiple cell types, differentiated cells, adult stem cells, embryonic stem cells. And long story short, you would get different responses to the scaffolds depending on these biological cues. So again, that was sort of trying to understand the basics of cell interactions with materials. But how do we translate this to the clinic? Now, we did not want to worry about delivering cells. It's a real challenge even still today. And actually there's new evidence today, and something that we thought about even 20 years ago, are these cells really viable? And there's a lot that can happen with dead cells, right? So recent literature has shown how in cardiac repair, it's actually dead cells that's having the therapeutic impact. And we'll talk more about that in a little while. So we went and looked at our development partners. So what are the surgeons doing in the clinic already, and how can we integrate what we learned in the laboratory with cell material interactions to make something that can go in the clinic in a pretty efficient way? So when we were working in cartilage, microfracture was a very popular technique, a little bit less so now, but it's still used. And essentially a microfracture process is taking a little drill or hammer and essentially making these holes in the defect that's in the cartilage surface and into the bone and bringing up the bleeding. And I'll show you that in a minute. So this allowed us to use endogenous cells, so we didn't have to deliver anything. And it worked with a procedure that the physicians were already used to working with. So this is a picture of the process. So in order to get that hydrogel to stick in place, we built an adhesive. So again, that was a project with the surgeon and the chemist working together. But we put on this adhesive, which was based on chondroitin sulfate, which had nice biological activity. And then we do the drilling for the microfracture and then fill up that defect with the hydrogel, which is binding to that adhesive. And essentially your bleeding process in that microfracture, it gets trapped in that hydrogel, and essentially you're making that blood clot stronger. You're enriching more components from the marrow as the bleeding gets trapped in there. And also we're discouraging fibroblast growth. So it was not the same mechanism necessarily looking at how cells are responding in the dish, but it's guiding tissue development. And so there were two clinical trials of this technology. The first one lasted 1 year, and the second clinical trial was 2 years. And that was really critical and is actually a challenge in the field because essentially microfracture starts failing after 18 months, right? And that's a long time to wait if you think of running a clinical trial. But anyway, it's nice to see in our data, the red being the microfracture control and the green is microfracture plus the implant. So after 18 months, your microfracture starts failing, and when you have a hydrogel implant in there, you have a more robust cartilage formation. And also the quality of the cartilage is better. So this helped us understand that we don't necessarily need to follow the traditional paradigm of tissue engineering where the biomaterial serves as a three-dimensional framework. We were redirecting wound healing. Now, a little bit after that, we had another clinical trial that was in partnership with a company called Kythera. And I would often joke that this was our dark side project. And that's because it was working with tissue fillers for wrinkles. But we just designed a whole spectrum of synthetic biological composite hydrogels, hyaluronic acid and polyethylene glycol that were light activated, and you can manipulate the properties nicely. But what was interesting here is their clinical trial was essentially taking patients who were going to have a tummy tuck, saying, we'll give you a free tummy tuck, but in exchange, we want to make these implants 3 months in advance. And actually, this is what the FDA is requiring for looking at these materials. Now, this is actually a really unique opportunity because how often can we get an implant back that's been in a human for just a matter of months, right? So real opportunity to see what goes on. And what we noticed is that the same biomaterial had different responses depending on if it was adjacent to subcutaneous muscle, adipose, or dermis. So the same biomaterial, depending on what tissue is around, has a different immune response. And we often, might complain about reviewers' comments in publications. Well, one of the reviewers asked us to look for a certain cell type, immune cell type that I really didn't know much about. And we did that, but it really introduced me to the sort of complexity of the immune system. And so we've actually completely pretty much redirected our research now to understand the fundamentals of the biomaterial immune response and using this as a target to engineer repair. So actually this immune response, which can be very, very different in people compared to the models that we use in the laboratory or even large animal models before translation, is really much more complex. So, people, will have their history of infections, environmental factors, in addition to the genetics of their immune system that can really dictate and really impact that downstream repair. The other nice thing is that regenerative, that immunotherapies there are already a lot of drugs out there targeting the immune system. So it's an ideal target for trying to promote tissue repair. So we're now shifting to understanding the fundamentals of this immune response. So if we look at normal healing processes, after any injury in your body, you will get an immune response. And so we were interested how that fundamental immune response is when there is a healing tissue versus what does the immune response look like in a non-healing tissue, which is characterized by actually a chronic inflammation and fibrosis and scar formation. And can we use this information of how these two immune environments differ to help push that non-healing environment to look more like a healing wound? So the examples that I'll be talking about these two environments today are a muscle wound. So if you think about it, we're constantly damaging muscle. If you're exercising, hiking, running, you're constantly causing damage to muscle. Of course, if you have a big injury, that's going to be hard to repair. But in general, it's a tissue that can repair pretty well. Now, that's opposed to cartilage, which, as I showed you before, was the target of our early regenerative medicine approaches. Now, when cartilage is damaged, you generally get scar formation and a chronic inflammation that can lead to arthritis. So can we learn something from these two immune environments, or how are they similar, how are they different? So one important point then to think about is an immune response is not always bad. And actually it's required to repair tissues. It's the type of immune response that is important. And what's been really interesting shifting from stem cells to immune cells is that with stem cells, you look in the literature, people would throw 100 million stem cells, 200 million stem cells to try to build tissues. But in the immune system, a very small number of cells at the right place at the right time can have a huge impact. So it's a real change in sort of a focus of how we have to dig into understanding this immune response. So just a little background, I have 3 slides to sort of give a preview of what I'll be talking about later. So we can broadly categorize the immune system into the innate immune system and the adaptive immune system. So the innate immune system includes a number of cell types that you may be familiar with biomaterials, in particular macrophages and neutrophils are very well studied in the biomaterials response. Now, these cells tend to be fast responders to injury or an implant, but they don't have memory, and it's not very specific of a response. That's opposed to the adaptive immunity, which takes a little bit longer, but it is a specific response, responding to specific antigens, and there's a memory to it. So the CD8 cells are T cells that are active in directly killing cells, and so the cancer folks are very interested in these cells. I'll be talking more, though, about CD4 cells, and their main job, of these T cells is to secrete cytokines that can impact pretty much all the other immune cells. Now, these T cells are interesting in that they have a specific response. So the T cell has a T cell receptor, which is specific to a certain antigen that is presented by the MHC molecule. And then there are various checkpoints here that you may have heard of in the cancer immunology field that can either put on the brakes or activate these cells. So the environment in which these T cells are being educated to these antigens plays a big role in the subsequent response. Now, I'll be talking about a few different types of immune responses. And I think this is really where engineers can come into play, because you'll see that a number of different cell types all contribute to sort of the immune environment. So type 2 immune responses are associated with the production of the cytokines interleukin 4 and 5 and 13. I'll be mostly mentioning IL-4. Now, these responses are connected with host defense, in particular worms or helminths. They're also associated with negative things and problems like asthma and allergies. But there's strong evidence of their role in tissue repair, in particular muscle repair and liver repair and even central nervous system repair. Now, there are a number of different cell types that can produce it. So actually, eosinophils are a big source of interleukin 4, even in muscle repair. And then there are these interesting cells called innate lymphocytes. That sounds like a contradiction, because I said the innate immune system is one component, and then lymphocytes or T cells are in the adaptive immune system. So these are actually cell types that come from the same lineage as T cells, but they behave more like innate cells. So they're in the tissue and they can respond quickly to cytokines, but it's not antigen specific. And then of course we get to the T cell. So this is a CD4 T cell that's making IL-4, and that's called a Th2 cell. And then again, that's a specific response. So that's a type 2 immune response. Now we can also look at type 17 immune responses, and that'll be sort of the second half of the talk. Now, this is associated with production of interleukin 17 and some other cytokines. Sometimes it's called a type 3 immune response, but type 17 is a little bit more descriptive. Now, this is involved, again, in host defense, right? Everything is about host defense and getting rid of infections. But it's also associated with autoimmunity and fibrosis. In particular, things like idiopathic pulmonary fibrosis, or IPF, which is a huge area of research. And actually a lot of companies are looking both at autoimmunity and fibrosis. And if you look at sort of where a lot of technologies are moving in the autoimmune field, IL-17 inhibitors are a big area of growth in both small and large companies. Now, who's making IL-17? Well, there's some interesting types of T cells called gamma delta T cells, and they also sort of sit on the bridge between innate and adaptive. So they're T cells, so adaptive immunity, but they don't respond to MHC protein antigens. They can respond directly to antigens such as phospholipids. So sort of non-typical antigens, and they tend to be residing in the tissues. Innate lymphocytes, a different type of innate lymphocyte called an innate lymphocyte 3. Again, similar to the one I mentioned before, except it's making different cytokines. And then finally, the CD4 T cell, the Th17 cell, is making IL-17. So those work together to create the immune environment characterized by interleukin 17. Okay, now back to biomaterials and tissue repair. So I said that there is an immune response to trauma. Well, biomaterials can modify this immune response. And I'm going to broadly characterize these as biological and synthetic. So biological scaffolds being derived from the extracellular matrix of tissues, also called decellularized scaffolds, but I really avoid that term because as we looked at them further, you might not be able to detect DNA, but there's tons of intracellular proteins that are sort of really stuck to this matrix. And there's a whole field of immunology associated with intracellular proteins that can activate the innate immune system. So whether it be sort of pathways, viral infections, and just understanding damaged cells, those intracellular proteins are really key to immune responses. So they're not really decellularized tissues, so biological scaffolds. They're known to create a pro-regenerative environment that we'll talk about next. Now that's opposed to synthetic scaffolds. Now this might be a little bit of a stretch and some people might like to complain about this, but I'm going to broadly say the synthetic materials will always have some type of foreign body response. Even if it's a very small 1 or 2 cell layers thick, there's still going to be a capsule around those materials. So can we use that as a model for fibrosis as we're trying to compare those healing and non-healing environments? So that's going to be our model for pro-regenerative and pro-fibrotic tissue environments that we'll talk about now. So for the biological scaffolds, we first looked at a muscle defect. So muscle, as I mentioned before, repairs via a type 2 immune response. So interleukin 4 is critical for muscle repair. Now, Steve Badylak really pioneered the development of biological scaffolds and their application to muscle defects, even in clinical trials. So we actually made an array of extracellular matrix materials derived from different sources of tissue. This one's cardiac, but we screened a number of them. And you can see if you put that in this muscle defect, we get new tissue repair compared to a defect without treatment. You've got a gap of tissue that's missing here. Now, so we use this as a model to understand the immune cells that were there. So this is flow cytometry, but a t-SNE plot of flow data, just so you can see the colors and how they're different. And this is focusing on the myeloid populations, which are more the innate immune cells. So things like monocytes, macrophages, neutrophils, and eosinophils are all going to be in this coloring. So just to show you that the myeloid cells are very different. So different cells are responding to the scaffold versus no treatment. But we're interested more actually in the adaptive immune environment. There's always a crosstalk between the innate and the adaptive. And we sort of like to think of the adaptive as being an orchestrator, right? So depending on what those adaptive immune cells are doing, they're going to be directing a lot of other cell types. So if we look at that wound and implant, we see that the percentage of CD4 T cells increases with scaffold, but we didn't know what kind. So we can look actually at what these T cells are secreting. So those same cytokines that I just mentioned to you, adding in one more, this type 1 response for interferon gamma, we can compare all of those and understand who is the dominant one in there. So a short summary is that after screening all of them, we found that interleukin 4 producing T cells were really important for scaffold response. So in a regular wound, muscle wound, you have a number of T cells making IL-4. You have a lot of eosinophils also making IL-4. But when you have that biomaterial in there, you essentially get a power boost of that IL-4 response. Now, what does that do to other cell types? Well, macrophages are well known to be important for biomaterials and tissue repair. Now, at the beginning here, I'm just going to talk about CD86 and 206, but later we're going to get a better definition of that. CD86 is pro-inflammatory, considered pro-inflammatory, and the cell surface marker CD206 is considered anti-inflammatory or pro-regenerative. And what you can see is that the scaffold actually decreases CD86 without changing too much in 206 except for the collagen scaffold, which is a control that we added. Now, what happens when you don't have T cells around? So the rag knockout model is an animal that does not have T cells or B cells. And essentially you get a double whammy. You have increase in that pro-inflammatory macrophage, and you lose that CD206 expression. Right, so those T cells are important for putting the macrophages in the phenotype that we want. Now, a student in this was very astute and looked at the draining lymph nodes. So this is far from the implant, far from the wound, and noticed that when there was an implant present, the lymph node was larger. So we took out both the local and the distal, local draining lymph node and the distal lymph node, and looked for those cytokines that I mentioned before. And we actually see increased production of expression of IL-4 when you have a biomaterial implant there. And this is important in a number of ways. In my mind, what I think is most important is it shows how both the wound and the biomaterial implant could potentially have systemic repercussions. Right, so it's not just the local implant region, but actually systemically. Now, final with this, there's a protein complex that is important for, it's a metabolic protein complex that's important for differentiation of T cells to the Th2 lineage. And this protein here, Richter protein, is critical for that TORC2 complex to undergo Th2 differentiation. Now, if we take those rag knockout animals that I showed over here that don't have T cells or B cells, we can transplant them with T cells that are normal and healthy, and we can transplant them with T cells that are defective in that Richter protein and cannot undergo Th2 differentiation. What you see in the tissue that's produced is that when the T cells are normal, you get nice new tissue production. But if your T cells cannot, if they don't have that Richter protein and cannot differentiate, you get more fibrosis, you get adipose, you still get some tissue produced, but it's not the quality of tissue that we want. The animals can't run as well, the macrophage phenotype is different, it's just dysfunctional healing that's happening. So this is sort of an overview of what we think the biological scaffolds do and the role of T cells in that biological scaffold response. Now, what about that non-healing wound? So what does the phenotype of that look like? After I go through this, I'll stop with some questions or give some time for questions. So when you think of the history of the field, we're always trying to push the gas pedal on regeneration, right? We're putting in stem cells, we're putting in growth factors, we're trying to grow the tissue. But what about how there might be breaks in the environment? So if you think of your car and, you know, a good car with good brakes, no matter how hard you push that gas pedal, you're not going to move anywhere if the brakes are stuck, if your emergency brakes are still on. And to keep that in mind as we go through this next portion. So I showed you how we can rebuild cartilage defects in sort of a healthy environment. But what about when there's inflammation? And how does that sort of simple defect that we might have here lead to sort of a full degenerative process? Well, the same group that we worked with, the same company that we worked on with that wrinkle failure, is now working in the aging. And they're particularly interested in the area of senescence. Now, senescent cells have been found in human arthritic tissue for a number of years, but the implications of what that meant was really unknown. So what are senescent cells? So you're probably familiar with senescent cells if you're growing tissue in the lab, and you're growing cells in the lab and they just poop out, right? They're no longer proliferating. Like, okay, I'm gonna throw them out. They're no good anymore, even adult stem cells. And this is replicative senescence. It's connected with telomere reduction and cells just getting old. You can also have senescence through damage, so oxidative damage, such as gamma radiation, genotoxic damage, oncogenic damage. So senescent cells are considered to be a protective mechanism to prevent tumor cell proliferation. But what's interesting about these cells is not just that they're not proliferating, right? So part of the definition of senescent cells is they're in growth arrest, they're not proliferating. But what really defines them was the discovery of the senescence-associated secretory phenotype. So this SASP is connected with their negative effects on age-related diseases, chronic diseases like diabetes, heart disease, and you'll see soon arthritis. So the SASP is also connected with tumor production, but the tricky part here is that senescent cells are also part of normal development and they're required for tissue repair. So this is an area of really active research that's quite fascinating. So sometimes these cells are good, but they accumulate with aging and are associated with pathologies of aging. So some of the markers I'll be using that the field developed to define these cells are P16. So P16 and actually P21 are considered markers of senescence. They're usually beta-gal positive. And then you can look for SASP factors. So when you think of the application of senescent cells in wound healing, you have some type of damage that induces senescence. And senescent cells secrete their SASP, which induces recruitment of immune cells. Now, in a normal healing wound, these senescent cells get cleared and your immune cells will go away. Now, if you have some environment that prevents those senescent cells from clearing, you can get chronic inflammation and fibrosis. So we looked in the joint using a mouse model developed by Judy Campisi. In this mouse model, you have the P16 promoter connected with luciferase, so we can watch when those cells come up with bioluminescence. And there's also a kill switch, so when we deliver ganciclovir, we can induce apoptosis in cells that are P16 positive. So we did a sham surgery where we open up the joint, and then the model that we used in the mouse was an ACL injury. So we transect that ACL, and you can see that senescent cells increase after that injury. Now, if we deliver ganciclovir, which is that kill switch, the number, you see there's less bioluminescence, fewer P16 positive cells. And if we validate that with P16 gene expression, we can see that it increases, reaches a peak at 2 weeks, and then goes down with the normal injury, but then with ganciclovir, it goes down even further. Now, we waited a little while to deliver the ganciclovir because we didn't want to impede the normal wound healing process. So where are these senescent cells? Well, if we look at P16, it looks like they're on the surface of cartilage in young animals. But some of the key factors that these SASP is making are important cytokines connected to the immune system, including interleukin-6 and interleukin-1 beta. Now, if we get rid of those senescent cells with a genetic model or some drugs, otherwise known as senolysis, we can reduce the SASP, and this actually leads to reduced pain and increases tissue repair. So here's a joint that is injured with that ACL transection. And you can see there's a loss here of the orange staining, which is the proteoglycans found in normal cartilage. And you can see an irregular surface. Now, if we perform senolysis and remove those senescent cells, you can see more of that orange staining, and you can see a nice cartilage surface. Now, you can see reduced inflammation of IL-1 beta, and using a drug that is supposed to selectively kill senescent cells, decrease in SASP factor IL-6, and reduce pain. Now, this is in phase 2 clinical trials now. So phase 1 was really looking to see if senescent cells were present in people and did it associate with areas of inflammation and severity of disease. And then phase 1 was looking at safety of the drug. And now phase 2 is looking at efficacy in some different concentrations. So those are available on the website with the company that's pushing that forward, Unity Biotechnology. But can we still repair when old? So when we tried that same senolysis regimen in older animals, we saw reduced inflammation and reduced pain, but no tissue growth. And actually, that tissue growth in the young animals was a surprise. So just by removing senescent cells, you could let repair happen. So that's really where the analogy of the brakes happens. There was a capacity to repair there, but there was something holding it back, the inflammation from that senescent cell. But what about when you're old? So this was actually another reviewer comment, right? So again, how academics can really help guide the academic peer review process can be really helpful. So the question was, what about age-related OA without an injury, right? Can senolysis still help? Well, this is sort of tough because aging animals and long studies, but we called up Jan van Derzen at the Mayo Clinic, and he published something in 2016 looking at a mouse model that did the same thing as the previous mouse model did. And they looked at heart disease, diabetes, kidney disease. So I called him up, I said, do you have any joints left from those animals? And lo and behold, he did. And this is what we found, is really amazing. So they started treatment with the kill switch on these mouse at 12 months of age for 2 weeks until death, okay? So this is with the senolytic treatment, this is without. This is beautiful cartilage, incredibly beautiful cartilage. And without treatment, like you can't even see where the cartilage would be, right? This is really, really incredible. So this led us to think, well, maybe it is possible to manage age-related OA. So when we look at the age joint, first of all, their cartilage looks terrible to begin with. These animals are 18 to 20 months old. If we make that ACL injury, it just looks even terrible. But we were able to find a regimen of both local and systemic senolysis with actually different drugs. I can get into that if anyone's interested. And we can actually get pretty decent repair. So actually some of these joints that have been damaged and then treated with the senolytic look better than the old joints before there was any injury. Now we went and looked at the immune profile, and what we found in the draining lymph node in osteoarthritis was increased IL-17, and actually both in the joint and in the lymph node, but different versions of IL-17. And when you give senolytic treatment, IL-17 goes down, okay? But at the same time, IL-4 is going up. So in the young animals, any regimen of IL-4 would, any regimen of senolytic would increase the IL-4. In the old animals, it was only the regimen of that local and systemic senolytic treatment that gave us that IL-4 boost that actually gave us this nice cartilage repair. So just like in muscle, you needed that IL-4 for repair, but it looks like that IL-17 was inhibiting it. And that actually makes a lot of sense from the immunology perspective. So again, removing the brakes that are inhibiting healing. Now, one final part in this section is we were quite interested then in the connection between senescent cells and these T cells. So we took senescent cells and cultured them with normal T cells. What we found is those senescent cells induced Th17 differentiation. So that type 17 immune response when we had TGF-beta present. If TGF-beta wasn't present, you went to interferon gamma. And then if we took Th17 cells, so T cells, CD4 T cells making IL-17, cultured them next to healthy fibroblasts, they became senescent, as demonstrated by P16, IL-6, and beta-gal staining. So there's a positive feed-forward loop between senescent cells and that IL-17 response that is putting the brakes on healing. So overall, sort of summary for that, tissue damage is going to be inducing both senescence and an immune response. And here you have a communication network between the SASP produced by the senescent cells, which includes a lot of immune factors, and then you have all these immune cells that are secreting cytokines that can be inducing senescence. So we're calling this an immunologically induced senescence, which is a little different than the proliferative and redox senescence. So maybe I'll stop there and see if there are any questions before I move on.
Thanks, Jennifer. Yeah, we do have some questions. I'll call on Paul Dalton first. So Paul, you can unmute and ask your question.
Jennifer, it's really, I had a general question, your experience with endotoxins and how you move them from biomaterials. Maybe implicate.
Well, he's breaking up a little bit, but I think we got the main gist of his question.
Yeah, so that's been really important. Actually, I mentioned that in the next session when we talk about synthetic materials. And we did do endotoxin analysis with these materials because we were worried about that. And it was below the limit that the standard FDA assay was looking at. But just because we're below that limit doesn't mean it couldn't be having an important role. Now here, with just the orthopedic injury, I think there's less chance with it. The biological scaffolds, with our GMP manufacturing, which I'll talk about a little bit later, we do look at endotoxin, but again, it's below the detectable limit. So, that doesn't mean it's not having an impact. Now, is that going to be something that can go directly to, what's the connection between that and the T cells is another question, but I think it's something we definitely have to look at.
Thanks, Paul. Annie Zemper also had a question.
Hi, I had a question just with regard to the previous subsection looking at interplay between macrophages and T cells, where you're showing the flow cytometry. We know, over time for a proper regenerative response, we need this sort of interplay between T cells and macrophages. And I really enjoy like the fact that you use a transplant with the rag, because I think that really shows the critical nature of the T cell response in that. Have you guys explored, so two things, have you explored at all looking at sort of the interplay between the requirement of the macrophage and if, so for instance, like in a Lysem-Cree DTR or something like that, where you can take out the max and look to see whether or not you get proper T cell recruitment and whether or not, so I guess what the dynamic interplay is there, first question. Second question is, have you looked at more than one time point, just because this is such a dynamic process and I wonder how it develops over a week or whatever?
Yeah, so for the muscle wound, we generally look at 1, 3, and 6 weeks, although we've been adding 3 days with some new therapies we've been looking at, and that seems to be much more interesting for the gamma delta T cells. I think we did, we might have done a 1 day, but I think 3 days is where we spent the most time. So, one other point, just even beyond the rags, because when we looked at the rags in the joint, it was a little bit more complicated. And what's interesting there is the innate lymphocytes can compensate for the loss of the CD4 T cells.
Wow.
So some collaborators had warned us that they were looking at IL-17 in cancer in the gut, that, they thought they got rid of the Th17 cells, but then all of a sudden the innate lymphocytes started making more IL-17 to compensate for it. So that's why I've been going to actually some of the cytokine knockouts. So I'll talk next about the IL-17 knockout, but then the IL-4 knockout was important for the muscle work. And actually we looked at the IL-4, for the IL-4 you do IL-4 receptor knockout, and we found that the IL-4 was necessary for the senolytic response. So there's a lot to figure out between that. So the peak of macrophages, as far as their phenotypic skewing, is more 3 weeks, although you can see strong differences at 1 week. But then the T cells, we tend to look at their phenotypes a little bit more at 1 week. And it does seem like there's this interferon gamma IL-17 coming early from the T cells, and then you have IL-4 taking over. And there's some interesting work about Th17 cells differentiating to Tregs. So, even the interplay between the different cytokines in the T cell response, in addition to the macrophage, I think is going to be an interesting space. So the approach we're taking now is really pulling everything out and I'll show in a little while the single cell, and just trying to do a combination of the single cell plus the flow cytometry to understand what's going on.
Thank you.
So the paper has the different time points. So to give you point you to
Thank you.
more of that. Sure.
Jennifer, I have lots of questions, but I'll save them for later so you can continue.
Okay. So now to synthetic materials, and this is there's beautiful work from the 1970s where the foreign body response was defined looking at protein adhesion, neutrophil response, monocytes and macrophages, and formation of foreign body giant cells with macrophages, multinucleated macrophages, and fibrosis. And there's some mention here of lymphocytes, but talking to some of the folks that were working on this back then said, well, when we saw T cells, we thought it was a contamination and threw it away. So T cells are a little bit tricky because they're not always right next to the material, and they're few and far between compared to macrophages and the neutrophils. But as you know, we're interested in those cell types. So we took samples from our surgical colleagues from breast implants. So frequently after cancer surgery, mastectomy, you have placement of a spacer, expander, and then it's swapped for a permanent implant. So there's an opportunity there to get tissue samples pretty regularly. And then, of course, there is the opportunity to get samples for, we've gotten samples from somebody who thought they had autoimmune syndrome, breast implant autoimmune syndrome, and then a punctured implant, and then some implants that become too fibrotic and have to be replaced. So we took these tissue samples from these implants and sort of did a full screen of what's there. And lo and behold, we did see a significant number of T cells. So we did some further analysis of that, both pulling out those T cells and just doing expression profiling. But here's a summary of a number of different implants. So we have CD4 T cells, and what we found is that they're predominantly making IL-17. And we had gamma delta T cells, which was a real surprise because it's actually a significant number of those T cells that are making IL-17. And this was consistent with all patients, regardless of the time point, when the implant came from, the age, and other clinical characteristics. So we went back to the animal model and we looked at, well, what is this IL-17 doing? And this is jumping a few steps, but hopefully for time, that's okay. And here, we're looking at IL-17 knockout animals, and what does that do to the fibrosis? So here's an implant, and I've got to tell you first that this is polycaprolactone, and we chose polycaprolactone, and we tested a whole range of them, but we chose that because it's been shown before to give a robust immune response, so we knew we'd have a good signal to see, because I think it's always less in animals than in people, and it's also in clinical devices, so it has clinical relevance too. So these are the particles. This is a macrophage and a fibrosis stain. And we did both the IL-17A cytokine knockout, and these T cells were making both IL-17A and IL-17F. So we had to do the receptor knockout too. And what you can see just visually is fewer macrophages and fewer cells making that alpha smooth muscle actin. And then in the paper, you can see the whole range, but these are just some various genes that we looked at in the knockouts and wild types connected with fibrosis, transformative growth factor beta, S100A4, and a number of different collagens. And just to show you, we screened a number of different biomaterials and we put them in different places. And you might change how much IL-17 is made, but it always is going to be in that direction, we think. So what about the connection between IL-17 and senescence, though? Because I just spent the whole time talking to you about arthritis. We saw the senescent cells first and then found the IL-17. So is that still relevant here? Well, we found P16 positive cells around these materials in the animals. And in the knockout mice, without IL-17, we see reduced senescence. If we go to the clinical implants and look for IL-17, and P16, we see the presence of both happening. And if you do a correlation between P16 and IL-17 expression, we see that there's a strong correlation there. So this presents us with potentially a new therapeutic target for the foreign body response. And we think you need to target both, actually, the senescent cells and also the cells that are making IL-17. So in this work, I apologize, this is quite busy, but the top here is more inflammatory, down here is more fibrotic. Here are the treatments. We did navitoclax as a senolytic, and then we also treated with navitoclax and the IL-17 neutralizing antibodies, both for A and F. So you see with both, we can really get rid of the senescence. We reduce IL-6 expression, and TGF-beta and all these various collagens and fibrosis markers goes down with the greatest reduction when you hit the senescent cells and a neutralizing antibody for the IL-17 and IL-17F. So, where are we going from here? Well, we're really trying to understand in more detail who is there, what are they doing, and who's talking to who. And, what we've shown gives us some idea, but we're really so far from understanding that. So what we're doing now is we're taking out individual cell types, we're sorting them out, and then doing more in-depth profiling with single cell. So hopefully these tissue maps will give us some more targets that are connected with these pathologies. So we use these model biomaterial environments, and this is an example of sorting out the macrophages. Now, this is already a little bit biased because I'm sorting out just the macrophages that we generally look at for the CD86 and 206, so they're more differentiated. In any case, these are the clusters we got. You can see we got a number of different clusters that show differential gene expression. And here's what it looks like. So with our synthetic biomaterial, it's a little bit simpler. We have two terminal clusters. This is pseudotime analysis. I can get into if people are interested. And we have this F1 fibrotic 1 macrophage, very simple interferon gamma production, sort of a nothing very interesting there. This F2 is what we found is the most interesting one. Production of IL-36 gamma and a lot of genes connected with, guess what, type 17 inflammation and autoimmunity. Then with the ECM material, we see this R1, which seems to secrete a lot of chemokines, and it's essentially mobilizing the immune system. And then R2, we actually sorted out and confirmed that it's a phagocytic macrophage. R3, we didn't get into too much, but let me just go with further with this 86 and 206. Now, R1 and R2, you see some differences between 86 and 206. F1, F2, you would not be able to differentiate with 86 and 206, yet they're very different macrophages. Now, if we look at type 1 and type 2 genes, sort of if you just look in the literature and classify gene as a type 1 and type 2, you can see, or an M1, M2 macrophage, it's pretty much random across these macrophage clusters. And remember that M1 and M2 was really heavily defined in vitro experiments, not really in vivo life. So what we did is we looked actually at surface markers that might define some of these populations. And we found CE9 and CD301B and MHC2 high helped us differentiate the F1 and F2 and R1 and R2. And then you can use flow cytometry and get the actual protein levels and see in your different urinary bladder matrix, polycaprolactone, or just the regular wound, how much of each one do you have and the kinetics of that. And you can also do immunostaining. So the breast implant that I talked about with the IL-17 related response, you see IL-36 gamma and CD9. So it looks like they're macrophages that are making that IL-36 gamma. So that just shows you how we can really start moving to better understand these cell types. We're now doing this with fibroblasts and a great complexity in fibroblasts. So I just have a few more minutes for two stories, so I might run through this relatively quickly. So again, going to our surgical colleagues for inspiration. We talked about breast implants, right, and how we might be able to reduce fibrosis there. But another thing that's happening in the clinic is using fat grafting or adipose of this patient to do breast reconstruction. So a number of patients will opt for this if it's possible for them. But there's some challenges with reproducibility of the surgical procedure, and also you get these calcifications and cysts that form when you move that adipose. So we developed, in partnership with the Armed Forces Institute of Regenerative Medicine, an off-the-shelf adipose. So we start with an allograft adipose, and we remove the inflammatory lipids and essentially process it mechanically and chemically so it has a feel. The surgeons have working with the surgeons to get a feel for the normal adipose they're used to working with, and make an off-the-shelf substitute that can really mimic that. Now, preclinical studies, if we have just fat grafting, you can see these cysts that form as compared to the off-the-shelf graft where you have new adipose forming and cellular infiltration and new tissue growth, but without this calcification happening. And you can see a difference in the color between the two. So we did phase 1 a safety trial, and we're particularly interested in cellular infiltration, markers for new vascular growth and cell infiltration. And also it's an opportunity to understand, are we getting that same Th2 differentiation that I mentioned was important for the biological scaffold response. And we're now in phase 2. We finished groups 1 and 2. The FDA has asked us to do a dose escalation. So dose escalation of volume. So 4 cc's, 10 cc's, and next will be 20 cc's. So this actually can work in conjunction with breast implants, or also for patients who have smaller defects like lumpectomies. So finally, in I guess the last 2 minutes that I have, I just want to end with something that I think, again, is a nice connection with translation and moving science forward. So back in 1986, it was mentioned that there was a connection between the wound environment and the tumor environment. The tumor was a non-healing wound. And I've just shown you how wounds, there's a whole spectrum of them and whole different types of environments of those wounds. So now we can really look at that with a lot more accuracy to try to define those tumor microenvironments and what it might mean for treatment. But from the practical side, we had a company come to us and say, well, people are taking out tumors and putting these biological scaffolds that are pro-regenerative in that resection site. We want to make sure we're not somehow stimulating tumor growth, right? Does tissue repair, enhancing tissue repair, somehow enhance tumor growth? So we did this work, and we actually, this is with urinary bladder matrix that is sold by ACell. We tested a number of different tumor models. And not only does it not increase tumor growth, it actually delays tumor growth. So very counterintuitive, because a type 2 immune environment in tumors is generally with a negative prognosis. So we looked a little bit further, and as you might expect, a biomaterial increases T cell and B cell infiltration. We also get these lymphoid aggregates with the biological scaffold. But when we pull out the macrophages and T cells from the tumor, or the tumor plus the wound, tumor plus the biological scaffold, we see significant differences in gene expression. And if you look just at macrophages and classical M1 and M2 genes, they're randomly up and down, right? So the type 2 with the biomaterial in the wound, a wound healing environment, is different than the type 2 in a tumor environment. Now, what does this mean for potential treatment besides not enhancing tumor growth? It's actually really exciting if we look at a dose response of tumor cells. So this would be an example of, let's say you do a resection, but there's some tumor cells left over. Well, all these animals are treated with checkpoint anti-PD-1. If you have the scaffold there, you don't have any tumor growth, right? Even with just a small seeding, and about half of the ones without the biomaterial grew tumors. Sort of a moderate response here where the biological scaffold improves response to immunotherapy. But then when you give a really high dose of tumor cells, you get even a bigger benefit of the biological scaffold improving responsiveness to immunotherapy. And actually now in these new trials, these new adjuvant trials, the response of tumors look like a healing wound. Right, so again, we're converging the tumor and wound healing fields. So I'm going to skip over this summary, and well, actually, maybe I'll just go through it quickly. The type 2 immune response, biological scaffolds, muscle wounds, type 17 and senescence, chronic inflammation, fibrosis around synthetic materials. And now where we're going is we're looking at the impact of age, infection, gender, and other diseases, and how it might be tipping the balance in one way or another. Putting the two fields together of immunology and regenerative medicine, we still have so much to do to map this immune response and try to understand how it impacts downstream things. I'm calling a stem cell downstream. And then finally, using this information to develop immunotherapies and this nice accessible target that we can develop drugs for. So in sum, I think translation teaches, and we need to look at the environment both locally, but also systemically. And then think about the brakes, not just the gas, right? We have a great capacity for repair, actually, even in an aged environment. We just need to remove the brakes. So with that, many people to thank, collaborators and students, and also my colleagues at the Translational Tissue Engineering Center. So with that, I'd be happy to answer any questions.
Thank you, Jennifer. That was amazing talk. Really great to see how far this work has come. I understand some people may need to run off for other meetings, but I do have a question I'd like to ask, and then I'll ask more later. Really interested in the senolytic therapies, the local and systemic therapies. What are you seeing if it's gotten this far yet in terms of the safety profile, in terms of the timing and the dose for those types of drugs.
Yeah, so Unity has first looked at applications where there's a local delivery, so the joint where you're doing an injection in that joint space, and then ocular applications where you're delivering it in the eye, which is, again, a little protected, non-systemic. So the Nutlen, the drug that we used for the local injection, has been tested clinically before, I think, for some cancer treatments, and there are side effects of thrombocytopenia. And so there's some chemists that are making some new drugs, these PROTACs, that need to be enzymatically cleaved before they, when they're in the cell, before they actually do their effect, so you don't have that side effect anymore. And then also being more specific of which cell cycle these target the BCL, navitoclax targets the BCL pathway, but which part of the BCL pathway, being much more specific in how they're targeting it. So one big issue is in the field is what exactly is a senescent cell?
Yeah.
And that actually is not fully understood. And so it's been really sort of a screening process to understand which drug can potentially selectively kill. And if we take navitoclax in the joint, it doesn't kill the senescent cells there, but Nutlen does. Why? We have no idea. And which cell types are actually senescent, we still don't know. So there's a lot to do in the field.
Lots still to do. Yeah. Well, thanks so much. I'll let everybody go. I know Jennifer's really kindly agreed to meet with our Women in Graduate Sciences group. So, and you got to get onto that meeting. So again, thank you so much for joining us today, and thanks to everybody for joining.